Good day, everybody. I'm going to talk about carbohydrates. Health professionals need a knowledge of carbohydrates, their functions, metabolism, and food sources so as to educate a patient effectively about the amount of carbohydrates intake according to his health problem. And this is important in diabetes mellitus. Chemistry of carbohydrates. Each carbohydrate molecule is composed of three basic atoms: carbon, hydrogen, and oxygen. The ratio of hydrogen and oxygen atoms in carbohydrate is 2:1, which is the same as in water. Each of these atoms can form a specified number of chemical bonds. Carbon forms 4, oxygen forms 2, and hydrogen forms 1. Each atom, according to the valency, has a characteristic number of bonds it can form with other atoms. Notice that in this simple molecule of ethyl alcohol, each hydrogen has 1 bond, oxygen has 2, while carbon has 4. Carbohydrates in food are divided into 3 classes. The monosaccharides, each molecule is composed of 1 simple sugar. Disaccharides, 2 monosaccharide molecules bonded together and compose each disaccharide molecule. It's a double sugar. And the polysaccharides, these are complex molecules. They are composed of many monosaccharide molecules bonded together. It's a complex sugar. The monosaccharides, the 3 monosaccharide molecules commonly found in food are the glucose, galactose, and fructose. All have the same chemical formula, which is C6H12 and O6, but they differ slightly in the geometrical arrangement of their atoms, whether they are dextro or levo, and so on. The glucose is found free in food, such as in honey and fruits. It's the major building block of disaccharides and polysaccharides. Glucose is transported by the blood to the cells where it is oxidized for energy. Its normal concentration in the blood is between 70 and 120 milligram per deciliter. The galactose is not found in nature. Like glucose is present in nature, but galactose is rather a part of the disaccharide lactose. Milk is the primary food source of galactose. The mammary tissue in human breasts converts glucose to galactose and then synthesize lactose. Galactosemia is a rare genetic disease where an infant lacks the enzyme necessary to convert galactose to glucose. The result is that galactose in the body increases and accumulates to toxic levels, possibly resulting in manifestations like mental retardation, liver damage, and cataracts in the eye. Treatment requires that all forms of milk and lactose, which is the source of galactose, should be removed from the infant's diet. Fructose is found in many fruits as well as in honey. It is also a component of sucrose. Fructose can be absorbed into the blood from food, but the majority of it will be converted to glucose in the liver. Fructose is the sweetest tasting of all monosaccharides. A condensation reaction is a reaction by which we form a disaccharide from 2 monosaccharides. We condense, or 2 simple sugars are condensed, water gets out of this reaction, and we get a disaccharide. Like in this example, 2 glucose molecules condense, water comes out, and we get a disaccharide maltose. The opposite is hydrolysis, where by introducing water into a disaccharide, it splits the disaccharide maltose into 2 monosaccharides, into 2 glucose molecules. Disaccharides. A disaccharide is 2 monosaccharides bound together. 3 disaccharides are commonly present in food: the sucrose, formed of glucose and fructose, the lactose, composed of glucose and galactose, and finally maltose, which is composed of 2 glucose molecules. The sucrose occurs in nature in food, such as in sugarcane, bananas, and sweet potatoes. Table sugar present in the market is sucrose. Sucrose comprises 15 to 20% of the total calories in our daily food intake. Lactose is found in cow's milk and can be synthesized by women during lactation. Lactose in cow's milk is the energy source for bacteria that converts it to lactic acid, which is the source of the different taste in spoiled milk. There is a disease entity called lactose intolerance, which is the inability of some individuals to produce the enzyme lactose, which is necessary to digest lactose into glucose and galactose. This condition results in passing undigested lactose into the intestine, which in turn absorbs large amounts of water that causes GIT symptoms like bloating, abdominal discomfort, nausea, and vomiting. The maltose is not found in nature. The maltose in beer is produced by partial digestion of starch. Polysaccharides. A polysaccharide is composed of many monosaccharide molecules. It varies from several hundreds to over 1 million. 3 polysaccharides will be considered in this presentation: the glycogen, cellulose, and starch. First, the glycogen is a branch-chain polysaccharide composed of hundreds of glucose molecules. Glycogen is synthesized and stored in the liver and skeletal muscles. Approximately 100 grams of glycogen are stored in the liver and 200 grams are stored in skeletal muscles. Glycogen is an important source for glucose for the blood whenever the level drops too low, whenever there is hypoglycemia. Cellulose. Cellulose is composed of many glucose molecules but is different from glycogen in that it is a straight-chain polysaccharide. It is found in plants where it provides strength and support to the cell walls. The desirable dietary fiber intakes ranges between 25 to 50 grams per day. What happens to the cellulose, to the fibers in the body after ingestion? In the mouth, the mechanical action of the mouth crushes and tears the fibers in the food and mixes it with saliva in order to facilitate swallowing. In the stomach, fiber is not digested and it delays gastric emptying. In the small intestine, fiber is not digested and also it delays the absorption of other nutrients. In the large intestine, most fiber passes intact through the digestive tract till it reaches the large intestine. And here the action of the colonic bacteria, the bacteria flora normally inhabiting the colon, digest the fibers. Some fibers, by the bacterial enzymes, are converted into fatty acids and gas. The fibers have got the property of holding water, and this can help regulating the bowel activity and treating constipation. And it binds substances such as bile, cholesterol, and some minerals, and then carries them out of the body. Fibers in selected foods. We can find in the first food group, the bread, cereals, rice, and pasta contains or provides about 1 to 2 grams per serving. In the vegetable group, most vegetables contain about 2 to 3 grams per serving. In the fruit group, the fresh, frozen, and dried fruits have about 2 grams of fibers per serving, while legumes, many legumes provide between 6 to 8 grams of fibers per serving. The third type of polysaccharide is starch. Starch is similar in structure to glycogen in that it is a branched polysaccharide molecule composed of glucose units. However, starch is found only in plants such as seeds, cereals, grains, fruits, root, and in nuts. Starch is the major source of carbohydrate and total energy intake in many countries. Functions of carbohydrates. First, it is the source of energy, the main source of energy to the body. Each gram of glucose can yield 4 kilocalories of energy when metabolized. The brain tissue normally uses glucose as a source of energy. Therefore, a low-calorie, low-carbohydrate diet can impair the normal functions of the brain. Synthesis of other compounds. Glucose can be converted into other molecules such as non-essential fatty acids and non-essential amino acids whenever their levels are inadequate. Digestion and absorption of carbohydrates. In the mouth, starch digestion begins by the enzyme salivary amylase, which splits starch into smaller fragments, disaccharides and dextrins. Dextrins are small polysaccharides. These fragments are swallowed and passed by peristalsis down to the stomach. In the stomach, the hydrochloric acid denatures the salivary amylase, leading to no further digestion of starch fibers, leading to the delay of gastric emptying, thereby providing a feeling of fullness and satiety. In the small intestine, the rhythmic contractions of the stomach will move the disaccharides, maltose, dextrins, and other into the duodenum. The pancreas secretes the enzyme pancreatic amylase into the duodenum, breaking down the dextrins into maltose. Disaccharide digestion begins at this point. The intestinal glands secrete maltase, lactase, and sucrase enzymes, which digest the respective disaccharides into their monosaccharides. Absorption. Peristaltic contractions move the monosaccharides into the jejunum, which is the second part of the small intestine, where digestion is completed and absorption begins. Absorption is increased as a result of the intestinal villi projecting into the lumen. Each villus contains a blood capillary into which the monosaccharides pass via diffusion and active transport. These are the villi, which increase the surface area of the intestine to facilitate and increase the rate of absorption. The glycemic index is a method used to classify foods according to their potential to raise the blood glucose. We classify foods into low glycemic foods, which are ideal to consume, and moderate glycemic foods, which we should use with caution, and high glycemic foods, which are those foods that have the ability to raise the blood sugar very high, and this should be avoided. Metabolism of carbohydrates. Glycogenesis and glycogenolysis are two opposite reactions that interact to maintain a normal blood glucose range. In other words, the liver tissue alternates between anabolic and catabolic reactions with carbohydrates to regulate the blood glucose level. Catabolism of glucose. It's important as it results in the synthesis of the high-energy compound ATP. Two main processes are involved in the oxidation of glucose to form the high-energy ATP: the anaerobic oxidation glycolysis and the aerobic oxidation through Krebs cycle. Anabolism of glycogen from glucose is by the liver and muscle cells, and this is helped or brought about by insulin to form glycogen, which is called glycogenesis. It occurs whenever the blood glucose is high. The normal blood glucose level is between 70 and 120 milligram per deciliter. Whenever this level is exceeded, we have hyperglycemia, and the body normally corrects this hyperglycemia by glycogenesis. Whenever the blood glucose level drops below 70 milligram per deciliter, there is a hypoglycemia. The body corrects this hypoglycemia by the breakdown of glycogen with the aid of glucagon and epinephrine that stimulate the liver cells to undergo glycogenolysis. Thank you very much.